Hello and welcome to AlzeCure Pharma's Capital Market Day. My name is Ludwig Sjöström and I'm your moderator for the day. If you have any questions for AlzeCure, you can send them in via the forms on the right. And with that said, I hand over the word to the CEO Martin Jönsson.
Hello everybody and thank you for attending today's market update with AlzeCure Pharma. Today we will have several well-renowned presenters. We have the pleasure to have Professor Bengt Winblad from Karolinska Institute. We have Johan Sandin, who is PhD and our Chief Scientific Officer and one of the co-founders of AlzeCure. We have Pontus Forsell, our Head of Discovery and Research, PhD, also one of the founders of AlzeCure. And then you have me, Martin Jönsson, CEO of AlzeCure Pharma. And looking at the agenda for today, we will give you a short overview, talk about what's our vision and our pipeline. Then we will go in and have a session together with Professor Bengt Winblad, who will talk about Alzheimer's disease and the development in the field, unmet medical need, and what we can expect. After that, we go into our project Alzstatin, our preventive treatment in Alzheimer's disease, and Johan Sandin will give you a background and talk about the evolution of the project. Then we go to NeuroRestore, our project to improve memory and cognition. And here we will have Pontus Forsell talk about the background of the project and also our new data supporting disease-modifying effects. And then in the end, we will have a panel discussion where we will look forward to answer questions that you have sent in. And then we will have a concluding remark and give you some take-home messages. So that is what the agenda looks like for today. Then we go in and give you a brief introduction to AlzeCure Pharma. We are a company focusing on Alzheimer's disease and pain, and we know these are areas of huge unmet medical need. As a company, we can be seen as a spin-out of AstraZeneca, who was in the field of Alzheimer's and pain, but left it in 2012. And then AlzeCure was founded in collaboration with Bengt Winblad, Alzheimer's Fund, initially as a research foundation. And then in 2016, the research foundation became a commercial research and development company. We are a very experienced team with competence from early drug discovery, development, target identification, development of molecules, preclinical work, development, and out-licensing. And we are based at Karolinska Institute, where we also were started. And today we have 3 platforms of small molecule candidates with first-in-class properties. We have Alzstatin, which is an innovative preventive and disease-modifying treatment against Alzheimer's disease to try to make sure that people don't develop Alzheimer's. Then we have NeuroRestore, which is a novel symptomatic treatment for cognitive disorders, where we are focusing on Alzheimer's disease. And then we have the platform Painless, where we have innovative project for osteoarthritis and neuropathic pain. And since 2018, we are listed at Nasdaq First North Premier Growth Market here in Stockholm, Sweden. Our business model is that we are a research and development company, and our intention is to out-license our projects when we come into early clinical phase. But we are open also to out-license earlier than that. And then based on the out-licensing to get income streams to continue our research and development company, projects. We are only working with small molecules. And small molecules, as you can see from this picture, are much smaller than, for instance, large biologics. And the reason why we are working with small molecules is that small molecules have a higher likelihood of passing the so-called blood-brain barrier. And in the brain, our main targets are. But there are also other advantages with having small molecules. It's also that when you develop small molecules, you can also develop them into oral medication that can be used for home treatment, and also that you can produce the drugs to very low production cost. This is our pipeline of small molecule programs, and they are organized into our 3 different platforms: NeuroRestore, Alzstatin, and Painless. And if we look at NeuroRestore, we have had success and progress there. Last fall, we had a positive readout from a phase 1, where we showed safety, tolerability, and target engagement, showing that we are activating centers in the brain, which are central for both treatment of cognition, but also depression. Then in Alzstatin, we have 2 projects. And in Alzstatin, our preventive treatment against Alzheimer's disease, where we have so-called gamma secretase modulators, we this spring came with a new additional CD, ACD 680. And then in our Painless platform, we have 2 projects. And here we have ACD 440, a TRPV1 antagonist. And here we have a phase 2 study ongoing. We have had last visit completed, and we have received all our data and are now analyzing the data and expecting a readout latest this summer. And then we have TrkA-NAM, a negative allosteric modulator focusing on osteoarthritis. And today we will only focus on our Alzheimer's project. And what is so positive coming to the Alzheimer's field is that we have really seen progress and increased activity in the field of Alzheimer's disease. And this has truly been needed. With new data, the so-called amyloid hypothesis has been validated. Several antibodies have done that. And we have had positive outcomes in clinical trials, including in phase 3. We have a new product which has been approved in the US, lecanemab. They have done a fantastic job, and we look forward to see their success in the field. And we can also say that what's also been so positive for the field is that we have had ways to improve biomarkers in diagnostic, which are so important for the development work in the field of Alzheimer's, to really find the right patients. So we treat the patients and include the patient that really should have the treatment. Then, based on all the success we have seen in the field, we now also see additional big pharma companies entering the field, or even re-entering the field. Here we have MSD, we have Takeda, and we actually also have AstraZeneca, who have re-entered the field. And what we're also seeing is that much more investments are done now in the field. We have private equity investments, we have investment funds, which are coming into the field, investing in companies and projects. So a very positive development. And in the field of Alzheimer's, AlzeCure has 2 projects. We have Alzstatin, which is a disease-modifying preventive treatment against Alzheimer's disease. And we have a so-called gamma secretase modulator, which is targeting the amyloid production, decreasing the production of A-beta 42. And here we are developing an oral small molecule. And then we also have NeuroRestore, which we will talk about today, which is a TrkA-PAM, which should improve cognitive function. And with that, we mean to improve learning and memory capability. And also here we are developing an oral medication. And initially, we saw this as a symptomatic treatment, but with new data, we also see potential disease-modifying abilities. And with that, I look forward to hand over to Bengt Winblad, who is here to give an update on the field of Alzheimer's disease and talk about what is happening and where things are heading. So Bengt, please.
Thank you, Martin. I'll just take a share. Thanks a lot for introducing me. I'm Bengt Winblad, and I'm a clinician originally. I'm former clinician, I would say. I haven't seen patients the last 10 years, but I've been very much involved in drug discovery the last time. And before that, I was heading a big geriatric clinic and also a clinical trial center. I will give you an introduction. And how do I go this? First, I'll talk a bit about the societal impact of the disease. It's, of course, enormous due to the large number of patients getting the disease. And these are some figures from Sweden that I've taken. You can see that we total have 150,000 with dementia in Sweden. Most of them are having Alzheimer's disease. And in addition to that, we have many individuals having pre-stages of dementia. We call them mild cognitive impairment, and I will come back a bit to that later. And of course, the costs for our society are enormous. It's the most expensive disease we can speak about. And the latest figures here are that it costs Sweden, with an inhabitant number of 10 million people, 80 billion Swedish crowns a year. And you have to say 10 Swedish crowns is roughly $1 or €1. Also globally, of course, it's an enormous problem. We see every year 10 million new cases. The last years also have brought a lot of new basic knowledge. We are better understanding the mechanistic ideas behind why the nerve cells are dying too early in the disease. And we have both, I would say, genetic and environmental factors. And you can see that to the left here, you can see the amyloid beta plaques and the neurofibrillary tangles. Actually, Alois Alzheimer, who discovered the disease and described these pathological changes in 1906 already in a meeting in Germany. He also added to that the inflammatory part and some other histopathological hallmarks. And we can see that to the right here, we can see that we have sporadic cases really dominate the field. So sporadic AD, we have up to between 95 and 99% of all cases, and the familial cases who are, of course, coming a bit earlier in the lifespan, within 35 to 65 years, we have roughly between 1 and 3% of all, I would say. And we know from epidemiological studies that the environmental factors where you can do a lot to help mainly are focusing on the cardiovascular system, but you see high cholesterol levels, high blood pressure, low physical activity, and of course diet has also an influence on the health condition of the individuals. Genetic risk factors are ApoE4, for example, increasing the risk really, and you see a large number of ApoE4 individuals in the clinical trials. This is a figure where we can see Alois Alzheimer depicted, is looking up from 1906 on what is happening. He looks very happy, I must say. He must have thought that we would have a treatment in 10 or coming 20 years after 1906, but it's actually not until in the 1990s we saw the first symptomatic treatment strategies coming up with the 3 cholinergic drugs, donepezil, galantamine, and rivastigmine. And the last approved molecule in Europe was memantine, that is an NMDA glutamate receptor antagonist, we could say, was approved in 2002 by the European authorities. And now we come to the right, to 2021 or 2023, where we see 3 new antibodies being approved: aducanumab, lecanemab, and donanemab. Regarding donanemab, we can say that it's pending approval, the process in FDA. Also very promising for me is to look on a slide by Jeff Cummings, where we can see different molecules in clinical testing phase. It's phase 1, phase 2, and phase 3. Phase 3 trials are used for approval of a molecule, of a drug, and you see that in the middle here, the middle circle here, and you can see we have biologics up to the, on the top, then we have the disease-modifying small molecules, and then we have the more symptomatic drugs to the left here. Of the disease-modifying molecules, we can see those that I mentioned, aducanumab, lecanemab, and also they mentioned solanezumab and gantenerumab. This slide was produced in 2021, so it's a bit old now. What is promising here is that we see an interest not only in beta amyloid, we also see an interest in affecting tau, affecting inflammation and other modes that could contribute to the start of the disorder. And it's also promising that we see so many small molecules in clinical testing phase. We heard Martin mentioning the small molecules really can be taken orally. They are small in size, perhaps a bit lipophilic, and will pass the difficult blood-brain barrier passage, and hopefully also pass into the nerve cells later. Why is it then important clinically with an early diagnosis? I think it's important both for the patients and for the relatives to understand the disorder and the problems the patients show. It's certainly also important for the society. But the most important part, in my view, is certainly that the patient gets access to treatment before the pathology is too advanced in the brain. He or she can take part in clinical trials. What we can see here in Sweden and at our clinic is that the diagnosis today is really put much earlier. When I came to Stockholm in 1987, I think it was, I would say that we put a dementia diagnosis 10 years later than we do today. And this is mainly due to the development of biomarkers including both imaging biomarkers, positron emission tomography, magnetic resonance, but also due to biomarkers, chemical that you can measure in cerebrospinal fluid, CSF, or in blood, or in tears, or any fluid of the body, I could say. And we can see here that if we have here the time for the dementia diagnosis on the x-axis, we have a pre-stage that we call MCI, late MCI, early MCI. This is a slide from 2010. And then we have a pre-symptomatic phase that could be, like, say, 20 years before the onset of dementia. And already then we can see two parts changed here. The blue and the green line, it's showing the cerebrospinal fluid measurements of amyloid beta, and the positron emission imaging part showing amyloid content. Later, we see an increase of symptoms that is increasing close to the diagnosis. We can see that the cognition is impaired, the memory function especially, the function, the ADL functions of the patient is impaired. And in parallel, we see an increase or a decrease in fluorodeoxyglucose measurements. Tau is coming a bit later, and we can see a continuous increase of tau with the disorder here, and then it levels off during the phase of dementia, or slightly increases, we could say. So this is a bit of a Swedish specialty, the blood and CSF biomarkers. I mentioned that CSF really, cerebrospinal fluid, really reflects what is happening in the brain, but it turns out that you also can use more cheaper methods or not so invasive methods, really to just look on the blood or plasma biomarkers and what damages they reflect in the neuron, in the nerve cells. You can see also biomarkers that reflect something happening in the vessel walls, or you can see inflammatory biomarkers, or you can see metabolic biomarkers like insulin, whatever. But the main focus is on biomarkers that measure effects in the synapses or in the long myelinated parts of the neurons. And here you can see the parts measured in CSF is, for example, A beta, it's phospho-tau, it's total tau, it's NFL, reflecting more the neurodegeneration, and its inflammatory parts reflecting the inflammatory measurement or the concentration of gliosis or astrocytes in the brain. And just recently, we have seen an increased interest in measuring blood biomarkers, and we have those that we traditionally could measure, and we also have the addition now lately of the glycans that are really seem more brain specific and really can predict when a patient will develop dementia in time years ahead of diagnosis, we could say. In the US, there is a big trend to look on different phosphorylation sites of tau in the measurement. Martin mentioned that we have seen some antibodies coming up, and I am only bringing some data from the studies on the latest two, lecanemab, that was really developed here in Sweden by Lars Lamfeldt, and donanemab, that was really developed in the US, and recently Eli Lilly came with a press release of a positive phase 3 trial. What we can see, we saw it already with aducanumab, was that these antibodies that are injected intravenously could reduce the amyloid burden in the brain. And in the phase 2 trial by BioArctic, that is a really nice study, I must say, with a large number of patients. They could see that with the two highest concentrations, either given every 4th week or every 2nd week, the highest concentration, then 10 milligram per kilo body weight. So a person weighing 100 kilos will have 1 gram of the antibodies injected bi-weekly in this green line. And you can see that really the decrease of amyloid in the brain, and they claim that 81% of amyloid positive converted back to negative at this highest dosage. This was used also for the, I would say, for the rapid approval of lecanemab by FDA, this phase 2 study, and they added the side effects part from phase 3 to come up with this rapid approval. Lecanemab people was then licensed to Eisai and later also to Biogen, have done a large phase 3 trial. It's called Clarity, and you can see that they used as primary outcome here global measurements of cognition and function, and it's called the CDR sum of boxes. And in the study, they could show over 18 months treatment that the placebo group declined faster than those that were on active immunotherapy. And they claim that this slowing of progression was 27% better than when you compared it to the placebo improvement. And if you prolong these curves, you can also see how long time, I would say, that you can keep these patients in this lower or better stage of the disease. And what was interesting for me was that the curves are divergent, but we have no idea if this will continue over the time, so we have to do more long-term studies. But it was also that all CDR sum of boxes domains like memory, orientation, judgment, community affairs, hobbies, personal care showed improvement. It favors lecanemab in these studies. So it's, I must say, rather strong data, and just now the antibody is left to EMA for eventual approval. And if it's approved by EMA at the end of the year, then it will be the local countries that decide and discuss the pricing or the reimbursement of this antibody. The last reported positive antibody was donanemab from, as I mentioned, Eli Lilly. And they had a press release a bit more a week ago, and we had a top-line results from their Trailblazer study. And during 8 months treatment, they saw around the same effect. They saw 35% less cognitive and functional decline. And it was, as you see, a large study with more than 1,700 patients in. On the same time, we have to observe the side effects in studies like this, because it's not without side effects. It has to be shown that they can develop a brain edema with headache and vomiting, etc. And here they had 30% around reported side effects with the microbleeds and around 20% with brain edema. Of course, then it's obvious to compare the data for lecanemab and versus donanemab. And I must say that there are differences in the study populations. You have different scales to measure the effects. And there was only one common scale, the CDR sum of boxes, but also the outcome of this scale is influenced certainly by the different study populations. So I must say, in summary, these differences make it difficult to properly compare the results from these two studies. And regarding the side effects, I would give the same reason. It's difficult to compare, to properly evaluate the reported side effects due to different populations, study populations. Still, for me, it remains a very positive feeling. These two studies really represent something new, giving us hope for the future treatment of AD. And most probably then combined therapy will come up, a combined therapy with a molecule that has some other modes of action affecting inflammation or metabolism or whatever. And it's an advantage if, as we heard, it can be a small molecule that can be taken per dose, is cheap, and will pass the different barriers. We'll later hear about the two Alzheimer platforms that AlzeCure are having. And it's first the Alzstatin, the gamma secretase modulators. We tested the gamma secretase inhibition, where it has more or less a total inhibition, and we had rather severe side effects. But now these modulators seem to be without severe side effects, and it's a very promising approach, I must say, to reach oral small molecule. And we also heard about NeuroRestore, and we'll hear more from the coming speakers on this, where we really, by paros, giving drugs orally, small molecules can influence the nerve outgrowths, the interaction between the nerves, and stay the synapses, affect the mitochondria. These are I've written symptomatic effect, but it's certainly also probably having a disease modifying effect with this mode of action. So if I may sum up this, we have reached much further by all these trials and by the use of more individualized care, that using the biomarkers. Really, we can select today disease subtypes that are much more biochemically, at least I would say, homogeneous, and we have a better effect on our treatments. And the treatments are still focusing on the main pathological hallmarks like beta amyloid, tau. We will hear more about neuroprotection. There are certainly a lot of other trials ongoing regarding Bricos, regarding inflammation, etc. Saw that in the slide by Jeff Cummings. And we also see non-pharmacological treatment strategies based on the risk inhibition, where you find risk factors mainly through the epidemiological studies. And for example, here you have the finger studies by Mia Kivipelt. So this last slide just shows my collaborators, and Anka Silas-Dominguez, Vesna Jellik, Lars Danfeldt, Anders Frimo, and certainly Gunilla Johansson. Thank you.
Thank you, Bengt. And in the next presentation, we will have Johan Sandin.
So thank you. So my name is Johan Sandin, and I'm a CSO at AlzeCure Pharma, and I will present AlzeCure's disease-modifying therapy, Alzstatin, to you. We'll start actually by looking at a section through the brain. On the right-hand side, you can see an Alzheimer's disease brain. On the left-hand side, you can see healthy brain. And what is quite apparent is the massive volume loss in the diseased brain, which is the result of massive neuronal loss in this area. Another key hallmark of Alzheimer's disease is the appearance of so-called amyloid plaques that are composed of the amyloid A-beta peptide. This was a key hallmark that Alois Alzheimer saw already in 1906, as we heard, and certainly it's a key characteristic for Alzheimer's disease patients. Now, if we look at A-beta amyloid, as we heard, this is a really early finding in the disease, as can be seen here on the graph to the left. And shown in red is the accumulation of A-beta amyloid in the brain. And this starts some 15 to 20 years before symptom onset. And it's obviously something you would want to affect very early on to avoid secondary and tertiary effects. Now, what is then, how is this amyloid then formed? So this animation will show you this. So in the cell membrane, there is a protein called APP inserted. And this protein is cleaved by two enzymes sequentially. First, by the beta secretase, here shown in pink, and then by the gamma secretase, here shown in green. The net result is the release of a small peptide called beta amyloid, which is quite sticky and over time and with increasing concentration starts accumulating into larger and larger aggregates, oligomers, fibrils, and eventually these large amyloid plaques that are so characteristic for the disease. Now, these amyloid aggregates are toxic to nerve cells in the brain so that they become dysfunctional and eventually die off. So what Alzstatin does, it's binding to the gamma secretase and changes how the gamma secretase cleaves the A-beta peptide so that shorter fragments are released, which are not toxic and do not aggregate. Now, if we look at this animation again, but as a picture, You can see here the cleavage of APP, as we saw in the animation, first by beta secretase and then gamma secretase, releasing the toxic A-beta 42, which is the building block of these amyloid plaques. Now, as we heard from Bengt, initially, the industry tried to completely block the gamma secretase enzyme. However, gamma secretase also cleaves other proteins, which are important for normal cell function. Therefore, this approach was not feasible in the clinic. The industry then went over to try to block beta secretase. Again, since beta secretase had also other proteins that were important, blocking enzyme activity completely was not a feasible way forward. The solution was to rather try to modulate specifically the cleavage of A-beta. And this is these are the gamma secretase modulators. So here, these compounds do not block enzyme activity, but rather specifically changes how the gamma secretase cleaves out the A-beta peptides, so that the shorter fragments are formed instead of these longer toxic forms. So this is important from a safety perspective. Now here, this slide shows you the classical so-called amyloid cascade, which generates these toxic and damaging amyloid aggregates. And as you can see, the A-beta is cleaved out, forming single monomers, which then aggregate into oligomers, fibrils, and eventually these large plaques. Now, if we look at the A-beta antibodies that we heard Bengt talk about earlier on, these antibodies bind and clear aggregated A-beta peptides, late-stage aggregates such as fibrils, protofibrils, and plaques. Alzstatin, on the other hand, which is a gamma secretase modulator, reduces the production of the initial building blocks, the A-beta 42, and thereby prevents the formation of all of these A-beta aggregates. Moreover, it also increases the amount of these shorter peptides, A-beta 37 and 38, that have been shown to display protective properties. And this has been shown by multiple groups, showing that these shorter fragments can actually inhibit the aggregation of the toxic A-beta 1 to 42. Also, that these shorter fragments seems to correlate with a lower risk of developing the disease. Moreover, that they also can be used as biomarkers for Alzheimer's disease dementia. Now, if we look at Alzstatin, for example, ACD680 as shown here, we see a potent reduction of toxic A-beta 1 to 42 in cells. So with increasing concentrations of the compound, we can see a decrease in A-beta 40 and 42, these toxic species, as shown here in blue and black. At the same time, we see an increase in the shorter protective peptides, A-beta 37 and 38. The net result, however, is we don't affect the total amount of A-beta peptides, but we just change the ratio of the toxic versus the protective species. And this is also an advantage, because if A-beta does have a physiological role, we are not affecting the total amount of A-beta. We just decrease the toxic forms and increase the more protective species. So in fact, we are doing just the opposite to what most familiar or inherited forms of Alzheimer's disease do. Now, if we look at the effects of Alzstatin, such as ACD680 in vivo, this figure here shows the effects in animals, where we can see that a single dose of Alzstatin decreases the amount of toxic brain A beta 1 to 42 by more than 60%. So it's a potent reduction. As we talked about before as well, we don't want our Alzstatin compounds to affect the cleavage of other important proteins. And the picture here to the right shows 4 different groups which represent other proteins that are cleaved by gamma secretase, such as Notch. Now, as you can see here in green, that's a gamma secretase inhibitor, so blocking the enzyme activity completely. That also blocks the cleavage of these other proteins, while a gamma secretase modulator, Alzstatin, shown here in blue, does not affect the cleavage of these proteins. So it's specific for A beta, which is, again, really important from a safety perspective. So now, how is Alzstatin then expected to differ from the antibodies that are coming to the market? Well, first of all, as we heard, this is a small molecule therapy, which generally passes much more readily across the blood-brain barrier to reach its target site, the brain. And we've seen this, for example, with NeuroRestore, where more than 37% of the unbound compound actually pass across the blood-brain barrier, to be compared with about 0.1 to 0.2% for the antibodies. It also provides a more cost-effective treatment for chronic use than biologics, given much lower production costs. It's also highly suitable for early treatment, before the brain becomes heavily damaged and the patient develops cognitive symptoms. It's also suitable for home treatment, given that it can be taken as a tablet or capsule. So you don't need to go to the hospital to get infusions of the drug, which also decreases for society. Moreover, we're not expected to have the side effects that are characteristic of the antibodies, such as brain edema and brain microbleedings, so-called ARIA. Again, decreasing costs because we don't need to do regular brain scans and hospital visits to follow up on these so-called ARIA effects. We also see an attractive profile when it comes to the clinical trials, because we can do clinical proof of mechanism and central target engagement already in phase 1 clinical trials in healthy volunteers. Here we could evaluate both safety and tolerability, but also to explore biomarker effects where we would be able to show central target engagement. We could look at both A beta 42 and 40, so a reduction of these toxic A beta peptides, while in parallel also assessing the levels of the shorter fragments A beta 37 and 38 are protected, and thereby establishing that we have reached the target and activated the target. We can do these measurements both in CSF and plasma, as there are biomarker kits available as well. Now, if we look at the target patient populations, we see 2 major groups. One, which is a preventive therapy, where the patients would be identified based on genetic risk factors and biomarkers. And this would be a standalone treatment before onset of symptoms and before any major neurodegeneration occurs. And there, the target would be to prevent the buildup of amyloid. And here, we could also look at patient groups which have inherited forms of the disease. That includes Down syndrome, which is suitable for proof of concept clinical studies. There is also another opportunity, which is with regard to a combination or maintenance treatment in patients with established Alzheimer's disease after initial plaque clearance provided by antibody treatments such as lecanemab and donanemab, for example. Now, if we look at the competitive landscape, there are less than a handful of competitors. We know that Roche has a compound in phase 1, And there's also another compound from University of California, San Diego, which is also currently in phase 1. With our compounds being in preclinical development, we think that we are quite in line with the competition. Of interest as well to note is that Pfizer have published data with an earlier compound showing that gamma secretase modulators can indeed display both potent efficacy and good safety in man. So to summarize, what we see are the advantages with Alzstatin. We can reduce the toxic amylogenic A-beta-42 production in the brain. While doing so, we also increase these shorter peptides A-beta-37 and 38, which have been suggested to have protective properties. The net result is we don't affect the total amount of A-beta. So if A-beta does have a physiological role, we're not affecting that. The mode of action is actually the reverse of most familiar Alzheimer mutations. And our compounds in Alzstatin, such as ACD680, as I showed you, can potently reduce A-beta-42 production in vivo more than 60%. And we can achieve proof of mechanism data already in phase 1 clinical trials. And we see potential for both preventive and maintenance treatment. So with that, I'd like to acknowledge and thank the team here at AlzeCure Pharma, our collaborators at Karolinska, Bengt Winblad, Lars Kärnberg, and also at Gothenburg University, Professor Henrik Zetterberg, Lotta Agholm, and Stephanie Freeworth. So with that, I'd like to thank you for your attention. Now leave the word to Pontus Forsell for the next presentation.
Thank you for attending this presentation. My name is Pontus Forsell. I'm Head of Discovery and Research here at AlzeCure Pharma. Today, I will present some new and exciting data of ACD856, describing results for potential disease-modifying effects. But before we dive into the results, I will give you a short background of neurotrophins and ACD856. So ACD856 is a small molecule, positive modulator of TRAC receptors. So it enhances the signaling of neurotrophins such as NGF and BDNF. So why are the neurotrophins such a good drug target? Well, they have been known for decades. NGF was actually discovered in 1952 by Rita Levi-Montalcini, who was later awarded the Nobel Prize for her discoveries around NGF. So they are really essential for neuronal survival and function. They play a very large role in learning, memory, and mood. So ACD856 is a molecule that we have developed actually from idea to where it is today, a clinical stage compound. It has been tested in a microdose study where we addressed the pharmacokinetic of the compound. We have also studied it in a phase 1 single ascending dose study and in a phase 1 multiple ascending dose study where we addressed the safety, tolerability, and target engagement. So these studies demonstrated that we really have an excellent pharmacokinetic property of ACD856. It has a very good bioavailability. It was safe and well tolerated in healthy volunteers, and we had a really good blood-brain barrier permeability. Also, quantitative EEG demonstrated we had target engagement in the CNS. So the role of neurotrophins like NGF and BDNF in combination with the mechanism of action of ACD856 really implies that this compound could have disease-modifying effects. And I want to show you some of the mechanisms that ACD856 possess. So as I mentioned, it is a small molecule positive modulator. So the compound binds to the receptor of the neurotrophins, thereby enhancing the kinase activity and enhancing the signaling of these neurotrophins. So this enhancement can have both short-term effects, which we think are the symptomatic effects of the compound, leading to improved memory and antidepressant-like effects. But it can also have long-term benefits. And these long-term benefits includes neuronal proliferation or increased number of neurons. It can have neuroprotective effects and improve neural outgrowth. It can also improve the communication between nerve cells by improving synaptic function. And finally, we have shown that it can have an improved function on mitochondria, which is the energy source of the cells. So altogether, these functional outcomes suggest that there is a potential for disease-modifying effect of compounds targeting the neurotrophic pathways. So today, I will show you some in vitro and in vivo data demonstrating potential disease-modifying effects of ACD856. And we will start with looking at amyloid beta-induced neurotoxicity. But before we go into the results of ACD856, I will give you a short background of the toxicity that amyloid beta causes in Alzheimer's disease. So some of the key pathological features in the Alzheimer's disease brain are amyloid plaques, of course, leading to synaptic dysfunction, axonal withdrawal, and later on mitochondrial dysfunction. So these can all be direct effects of the toxic amyloid beta, or it can be indirect effects due to increased neuroinflammation or lipid dysregulation. Nevertheless, these events are actually closely correlated with cognitive dysfunction that is so characteristic for Alzheimer's disease. So any preventive, protective, or restorative treatment will have a significant impact on the disease. And we think it could be very interesting and especially valuable if you can combine a symptomatic effect with a disease-modifying effect. So any preventive treatment will lead to a slower or a stop the aggregation of amyloid beta, leading to the amyloid plaque. So this can be done at any step here already from the cleavage of APP to the A beta by gamma secretase or by antibodies targeting any one of these steps. A protective treatment will make the nerve cells and the brain more resilient to the amyloid beta toxic aggregates or to neuroinflammation or to mitochondrial dysfunction. Restorative treatment is probably a longer-term effect aiming to restore damaged part of the brain. And all these three different ways of targeting the disease are actually disease-modifying treatments. And these treatments, disease-modifying treatment, will affect the slope of cognitive deterioration, which is the black line here. So disease-modifying therapies will slow down the cognitive deterioration, the rate of deterioration. Symptomatic treatment here in orange will increase the memory performance for a short time, and then the disease will progress. What we think is exciting with ACD856 that we probably have combined effects, both of symptomatic effects as well as disease-modifying effects, leading to that we will hopefully see improved memory and then a slower progression of the disease. So looking into a little bit more in detail about amyloid beta-induced neurotoxicity on a single neuronal cell, here depicted to the left. So this is a healthy neuron. A healthy neuron has many different neurites. So these are just projections from the cell body. In the upper part, the neurites are called dendrites. So these are the neurites receiving input from other nerve cells. And on the dendrites, there are small spikes called spines. And it's actually on these spines that the actual communication between different nerve cells are being formed in something called synapses. So in the synapses, the neurotransmitter are released as signaling substances between nerve cells. And it can actually be up to 30,000 of these synapses or these spines in each nerve cells, and up to even 10,000 synapses per nerve cell. So there is a lot of networking going on here. In the lower part, it's the axon of the nerve cells, and this is where the nerve cells transmit the signals to neighboring nerve cells. So the upper part receives signals, and the lower part transmits signals. With age, amyloid beta will start to aggregate and form toxic aggregates. These aggregates will, by themselves, cause toxic events or indirectly start a neuroinflammation. Over time, this will lead to the first event on the neurons called spine degeneration. And as you can see, there are less spines here, presumably meaning that the nerve cells will have less capability to communicate with other nerve cells in the surroundings. As the disease progresses, the amyloid plaques will be larger and more in numbers, leading to synaptic dysfunction, where the synapses actually loses contact with other nerve cells. And even later on, the neurites will degenerate, something called dystrophic neurites. They are fragmented, and now we have more or less lost all the communication capability of the neurons, leading to, at this last stage, the neuron will actually die. So over time, the neuronal function will decrease from a healthy neuron to almost a dead neuron with no capabilities of communicate. And the synaptic dysfunction, neural degeneration, and neuronal cell death are all hallmarks of this amyloid beta neurotoxicity. And we think that ACD856 could have a neuroprotective role here and keep the neurites from dying, keep the neurites to have a larger number of neurites and also larger number of spines. So we have addressed this in cortical neurons, where we have added amyloid beta 42 to induce a neurotoxic event. And then we have looked at the cells with microscopy and analyzed a synaptic protein called SNAP25. And in the image to the left here, you can see that the healthy neurons have a large network, and the brighter parts here is the SNAP25 protein, and it's localized both in the cell bodies, but also in neurites and in the network connecting the different neurons. If we add A beta 42 to these cells, you can see that the network more or less is disappeared. The cells loses the contact with each other. However, if we add ACD856 to these cells, we can see that we can protect the cells from the loss of neurites and retain much of the network between the cells. And if we analyze the number and length of these different neurites in these cells, we can see that A beta 42 reduces the length of these neurites. So the lower the bar, the shorter the neurites are. And if we add ACD856, we can protect these cells and keep the neurite length in a dose-dependent manner. So do we think that this is really a strong evidence of a protective effect of ACD856 against amyloid beta-induced neurotoxicity? Next, I will show you some data concerning neuronal growth and neurite outgrowth. So neurotrophins like NGF and BDNF are important for cell growth, neuronal growth, and neurite outgrowth. And there's a special kind of cells called cholinergic neurons, which are dependent on NGF for their survival and function. So we have studied ACD856 on cell number and neurite outgrowth in immature neuronal-like cells. So these immature cells, they don't have many neurites, as you can see here. So the green small areas here are cell bodies. But if we add NGF to these cells, they will start to form neurites. And if we add ACD856 together with NGF, they will form even more neurites, now extending a large area and starting to communicate with each other, perhaps. Also here, if we analyze the length of these neurites, we can see that ACD856 really have a dose-dependent effect, significant effect on the neurite length per neuron. And in the lower part here, we can see a graph showing that ACD856 also increases the number of neurons in these experiments. So we both improve the neurite length as well as the number of neurons. So this suggests at least a disease-modifying effect, but maybe also a neurorestorative effect, since we do see an increased number of neurons. So next I will show you some results of BDNF and the effects of ACD856. So BDNF is a very important growth factor in the brain. It stimulates neuroplasticity and synaptic function. And it's actually one of few molecules that is being synthesized in activity-dependent manner. So when we exercise or perform strenuous work, the levels of BDNF are increased in the blood, and some of this BDNF actually reaches the brain, and it could be one of the reasons for the well-being after training. And there are several lines of evidence, many scientific articles showing involvement of BDNF in Alzheimer's disease, but also in depression. There is a genetic differences in man called BDNF Val66 MET. It's a polymorphism, and it's associated with cognitive impairment and actually worsened amyloid pathology in Alzheimer's disease. So carriers of ApoE4, which is a risk factor for Alzheimer's disease, and BDNF Val66 MET have actually, when they are combined, these patients have increased amyloid beta pathology. So this genetic difference, the Val66 MET polymorphism, it reduces the BDNF secretions from nerve cells. And this leads to an increased rate of cognitive decline in these patients. But we can also see an increase of different biomarkers of the disease, such as tau and phospho-tau. And as stated here, this is actually one of very few genetic factors that is known to moderate downstream effects, such as cognition and biomarkers in Alzheimer's disease. So we have looked at the levels of BDNF both in short-term experiments, where we looked at the cortical neurons incubated with ACD856 for 7 hours. But we have also looked at in aged animals, where we have dosed ACD856 for up to 4 weeks once daily, and then analyzed the BDNF content in the brain. So to the left here, we see how ACD856 increased the levels of BDNF in cortical neurons already at 100 nanomolar. And if we analyze the brain of animals who has been administered ACD856 for 4 weeks, we can see that there is a significant increase of BDNF levels in these brains. So this suggests to us that ACD856 improves the BDNF levels in animals as well as in cell culture, and this could lead to improved synaptic function, improved memory, but maybe also to antidepressant-like effects. So going into a little bit more into in vivo data that we have done recently. So we have studied the effects of ACD856 in naturally impaired memory impairment in old animals. So these animals were dosed with ACD856 for 4 weeks, for one single dose, and then we performed 2 memory tests. So ACD856 was dosed once, and then the animals were allowed to learn a task. And 1 day later, the animals were tested how well they remembered this task. And then they were returned to their home cage, and 11 days after the learning task, we performed the second memory test. And as you can see in this graph here, the young animals remembered the task very well. So the higher the bar, the better the memory. The old animals had a trend to decreased memory already after 1 day. And this trend was actually ameliorated with ACD856. When we performed the second memory task, you can see that the old animals had more or less forgotten the original task that they were learned. The young animals remembered it very well. Interestingly, the old animals that was dosed with ACD856 remembered the task as well as the young animals. So this shows that ACD856 really improves age-induced memory impairment and to a level, at least in these animals, that is similar to young animals. We also performed many other cognitive tests in animals, And we can say that ACD856 actually improves both the learning process and the storage of memories and the recollection of memories. So all these three domains of memory formation is improved with ACD856. And we have also seen that repeated administration of ACD856, when we give it for 4 to 5 days, improves memory and presumably through a neuroplastic adaptation. So improving synaptic plasticity maybe, and this leads to a better memory. Finally, I would like to show you some long-term antidepressant-like effects that we have seen with ACD856. So here we have dosed animals once daily for 5 days, and then we have stopped the administration and waited for 3, 5, or 7 days before we did the depression-like test. And we have compared ACD856 to ketamine, which is a well-known antidepressant molecule, but it has some limitations which limits its use. So this graph here shows the effects 7 days after the last dose of 856. And the lower the bar, the better the mood. And we can see that ACD856 has more or less the same effect as ketamine 7 days after the last dose, suggesting that it is a long-term effect of ACD856 in this particular model. So to summarize this, we have shown several lines of evidence suggesting potential for disease modification. One is the neuroprotective effect against amyloid beta-induced neurotoxicity. Secondly, we have seen that ACD856 enhanced neural outgrowth and neuronal proliferation. We have also seen that ACD856 increased the levels of BDNF both in cortical neurons and in animals. So it improves memory, presumably via increased synaptic function, which can be mediated by the increased levels of BDNF. And we have, interestingly enough, demonstrated a sustained antidepressant-like effect lasting for up to 1 week after the last dose of ACD856. So this combination of short-term effect, the symptomatic effects leading to improved memory, and these presented an exciting long-term effect suggests that there is actually a new potential for disease-modifying effect of ACD856. And in conclusion, these preclinical data suggest a new potential use of ACD856. We think that this is very encouraging and supports that ACD856 actually can improve memory and learning and memory in multiple ways. The neuroprotective and neurorestorative effects are really add-on effects to the symptomatic effects that we have previously seen with ACD856. And to wrap this up, we think that these are really exciting data and introduce a potential for disease-modifying treatment to the already established symptomatic treatment of ACD856. And I'd just like to acknowledge the team at AlzeCure Pharma, our collaborators at Karolinska Institute, especially Professor Bengt Winblad and Professor Maria Eriksdotter. And with that, I thank you for your attention.
Thank you very much, Pontus. Great presentation. And now we are through today's presentation, but now we are coming to a very important part, and that is, of course, the Q&A session. And the Q&A session will be led by Finwire and Ludwig Sjöström. So I hand over to Ludwig. Please, Ludwig.
Thank you so much, and thanks for the very interesting presentations here. I think we will move on with the questions right away. And the first question is for Professor Bengt Winblad. Why do we see success in the Alzheimer's field now?
Yes, this is of course a question that I follow the long-standing basic research, even if I'm a clinician, and seen that we have understood much more about the brain function in aging and the brain function in dementia, especially in Alzheimer's disease. So it's a result of a long-standing basic research. I mean, we have only focused perhaps on amyloid. Now we also focus on tau, we focus on inflammation, and we can see that we have seen development of specific drugs having a more direct observation. I think we also have a success due to that we have selected patient populations better for the clinical trials. We have been more careful in getting homogeneous populations, and the development of the biomarker field has been really important, both in selection of patients, but also in measuring the outcome, I would say. So I'm really happy to see the success in the Alzheimer field the last 2-3 years. And of course, what we have heard today, we have much to expect of these more small molecules that will add effect or having an additive effect or being even used preventive or in long term. So it's a really interesting future.
Thank you. Here another question for you as well. Why is there a need for other treatments against Alzheimer's now where we can expect the antibodies to become available?
Yeah, that's I understand the question. It's of course urgent that we not only find the really expensive antibodies. It's expensive to develop antibodies, and they have also rather limited effect. And I can say that the cost effectiveness as the effect size is not so big compared to the price that they are asking for. So it's if you bring in health economic terms in this, I like the term cost effectiveness. Then you see that the effect size is not too big. I would also say that we have side effects, and we have really to find molecules that don't have the side effects and molecules that can be cheaper, but also having less side effects, and also that we don't need to do the intravenous infusions. I mean, the immunotherapy with antibodies is always done by intravenous infusions. I mean, so far the subcutaneous approach is of course promising, but it has still shown negative results. So I think both a question about specificity, about cost, and about lessening of the side effects is important.
Thanks. The next question is for Johan Sandin. What's the advantage with small molecule drugs versus biologics/antibodies?
So thank you. So that's primarily, I would say it's based on that small molecules as such. They pass more readily across the blood-brain barrier, given that they are small molecules, so they can be designed to pass the blood-brain barrier. If you compare to antibodies, which pass about 0.1 to 0.2%, as I mentioned, across the BBB, we see, for example, with NeuroRestore ACD856, more than 37% actually pass the blood-brain barrier. So that's one key feature. The other one is obviously that small molecules can be taken as orally, so as a tablet or a capsule, which makes it much easier to take at home. So you don't need to go to the hospital for intravenous infusions. So that's obviously a major advantage, especially for this group of patients where a hospital visit can be quite cumbersome. And also, if you look at the cost of small molecules versus antibodies and biologics, it's a major difference. The production costs for small molecules is much cheaper, so it would provide a much more cost-effective solution for the majority of patients out there. As we heard Bengt talk about before, there are some 55 million patients with dementia worldwide, so there's a huge need to fulfill with these type of drugs.
Thanks. I have a follow-up question for you as well. What's the advantage with a gamma secretase modulator such as Alzstatin compared to antibodies?
So that I would say is, first of all, we with the gamma secretase, we target the actual initiation, the formation of these small building blocks that actually form the amyloid plaques. So it's highly suitable for early intervention. Moreover, we see that we have this production of shorter A-beta peptides, such as the A-beta 37 and 38, which have been shown to display protective properties. Moreover, we do not expect to see any of the kind of effects seen with the biologics with regard to brain edema and microbleedings, so-called ARIA, which is a major side effect with the antibodies. And this is nothing we would expect with the gamma secretase modulators.
Thanks. The next question are for you, Martin. When can we expect to have efficiency data in humans with Alzstatin?
We actually expect that we should have efficacy effect data with Alzstatin already in a phase 1 in the multiple ascending dose study. So when you go into a phase 1, you normally only look for safety and tolerability, but since we, as we expect, will be able also to measure to what effect we can decrease the production of beta 42, which is the toxic protein which builds up to plaques, and then also to measure and look at how we can increase the production of A beta 37 and 38, which we think can also be protective. So we actually think that we can have efficacy data very early. So that's very advantageous, not at least from a business development or an out-licensing strategy.
Thanks. And the next question is for Professor Bengt Winblad. What is the unmet medical need regarding symptomatic cognitive enhancing treatment such as NeuroRestore?
Yeah, for me, NeuroRestore offers a completely new possibility to, via the oral administration with a small molecule, really stimulate the NGF or BDNF, the nerve growth factor system. Earlier, we had to use our neurosurgeons to operate in a catheter, then to put the catheter and in the tip having cells that produce NGF or BDNF and place it into the area of the brain where you produce, for example, the cholinergic acetylcholine chemicals. So I think this is a real step forward in treatment strategy to be able to use a small molecule peros that is having an acceptable price and having, as I understood, very few side effects, if any. It's unique, I would say. And perhaps also the indication prevention was discussed. I mean, can we use this on preventive strategies or other disorders like in Down syndrome, whatever? So I think this is an interesting approach, and it's many complications with other treatments that we can avoid, and so we have really much less side effects, and I hope the hopes will be fulfilled with this molecule.
And if I could fill into what Professor Winblad said, is that if we look at the antibodies are slowing the disease progression. But in these patients who develop Alzheimer's disease, you have an impaired cognition. So with a symptomatic treatment like with NeuroRestore, you have the benefit that you should improve memory and learning abilities. And this is also what the patients really are asking for. So in the surveys done with the patients and the family, the main thing they really ask for is really improved learning and memory capabilities. And that's why these symptomatic treatments, memory-enhancing treatments, learning-enhancing treatments are so important.
Yeah, I agree with you that as a medical doctor, I can see the ethical dilemma. Yeah. How do we inform our patients that they get immunotherapy and they are not getting better, but they are getting less bad, I would say. That will be a difficult task to convince patients to start the treatment. And this is it's promising if we can improve something. Yeah, I agree on that.
Yeah. So we need combination treatments.
Yeah.
Thanks. The next question is for Pontus Forsell. Why is the disease-modifying data potentially important for the NeuroRestore platform?
Well, I think it will be very important for the patients. So this will be a pill that can be taken at home, and the disease-modifying effects will probably slow down the rate of cognitive decline. And if you combine that with the symptomatic effect, we do have a combination therapy in one pill, actually, both a symptomatic treatment and a disease-modifying treatment. And that combination might also extend to other indications, not related only to Alzheimer's disease. And the second thing I would like to comment is, maybe it's been touched upon already, but it's a safe and well-tolerated molecule. So we hope it's amenable for long-term administration to patients.
I got another question for you as well. Which patient should be given NeuroRestore, and especially if the drug has disease-modifying abilities?
Well, if you look at Alzheimer's disease, we think that the patients that at least we will start to treat are those with mild cognitive impairment or mild Alzheimer's disease. But that can later on be extended maybe to early MCI, subjective cognitive impairment, maybe to moderate Alzheimer's disease, but as I mentioned, maybe to other indications as well, as we see a great potential in this molecule. I mean, we have seen antidepressant-like effects in animals, so that is something that we think are really exciting and interesting. And yeah, as I said, the long-term effects will probably be very beneficial for the patients and their quality of daily life at the end.
And if I can add to Pontus' comment, as Pontus alluded to, there are several other indications. We know that there are so many other patient groups who have cognitive deficits. You have in Parkinson's disease, you have in traumatic brain injury, you have post-surgery. There are so many different conditions where you have cognitive deficits, which potentially could be treated with regard with NeuroRestore. So very, very exciting.
Thanks. Another question regarding NeuroRestore for you, Martin. How can a disease-modifying data with NeuroRestore contribute to with additional opportunities regarding business development?
Very good question. Initially, when we started NeuroRestore, of course, we knew about the biology and everything that were, how should I say, alluding to that we could potentially have disease-modifying abilities. But initially, we only looked at the symptomatic effects, increasing learning and memory capabilities. But if we now also have these disease-modifying effects, this is a major step forward, because it could mean so much to the patients with regard to improving the neural health, synaptic health, and also increasing the mitochondrial activity. So to add this value to for potential out-licensing deals for other companies, it adds so much value. So if we really can show that we have something here with a disease-modifying ability, it's a major step forward. And then we also, as Pontus alluded to before, since we also have seen effects in preclinical model on depression, where we also have so many individuals, and we have such a big medical need also in depression. So if we can also show abilities there and have a very good, how should I say, safety profile, and hopefully also a quick onset of action, this could also mean a lot for NeuroRestore. So it means a lot, truly.
Thanks. Here's an open question for everybody, so anyone who wants to answer it is okay. How has it been historically for a beta amyloid project? Is in vitro effect a secure indication on in vivo effect, or do you expect that complex reactions or interactions may get in the way?
So I can take first on that. So with regard to beta amyloid, the whole amyloid hypothesis is something that has been worked on quite heavily during the last decades, I would say. And we've learned quite a lot. The picture showed earlier on showing the initiation of amyloid pathology starting some 15 to 20 years before symptom onset is actually quite a recent phenomena that came about when we were able to follow the aggregation of amyloid in the brain. And with regard to, we have also kind of got more understanding of how amyloid actually is being formed in both in cellular studies and also doing and inventing new, more predictive in vivo models of amyloid pathology. So I would say that the models we have right now, when it comes to kind of assessing the effects on A-beta 42, 37, and 38, both in vitro and in vivo have been quite predictable when going into human studies. So that was shown, for example, by the reference I showed earlier on by Pfizer, where they could see that, yes, the compound did what it should. It reduced A-beta 42 in humans, increased 37 and 38 as predicted by the in vitro and in vivo studies. So I would say that the field has come quite some way since the last years. And we also heard Professor Winblad talk about the advances made within the biomarker field around both diagnostic biomarkers as well as efficacy biomarkers. So I think this is really an exploding field right now in that specific area as well. So I think we're in a good position to move into clinical trials.
Thanks. Another question for you, Pontus. You mentioned potential antidepressive effects regarding ACD856. Do you see potential in conducting a study within that indication in the future, or is it that area already crowded with products?
I think there are ample opportunities for a new drug, a new antidepressant drug, actually. Some of the existing drugs have severe side effects. Not all patients can stay on the medication, and they have sometimes a long time to onset before you see an effect of the medication. So with respect to the antidepressant-like effects we have seen, I do think there is an opportunity for clinical trials within depression and ACD856.
Thanks. Also a follow-up question on that. Could the antidepressive effect be something negative if patients or doctor only want a neurological effect?
So what we have seen in animals, at least with respect to some models that we have tested, that ACD856 doesn't seem to have any effect if there is a stable physical mental state, there is no cognitive decline, then we don't see much of an effect of ACD856. So it seems like the compound needs to have that the system has been compromised in somehow, either by stress leading to depression-like behavior, or with age, like I showed you, you have a impaired memory. So I think the safety profile looks really promising. So it seems like the system needs to be compromised for the compound to have an effect in man. So we don't expect much side effects or off-target effects. And I think that many with depression also develop Alzheimer's later on, and vice versa. So it could actually be beneficial in both therapeutic areas.
Thanks. Now
maybe we can ask Bengt, do you see any potential negative effects?
No, I see most positive effects. I have more case observations of negative effects when you improve increase impression, but from studies I had in when I studied the severe stages of the disease.
Yeah.
And this drug is possibly effective in all stages of the disease.
Exactly.
I think you could even go on. There are very few trials on moderate and severe stages of the disease. And the health economic gains to have a drug on that would be fantastic.
Yep.
So I mean, to keep patients out of the nursing homes is a cost win in Sweden, I would say. And to have relatives spending fewer hours with these patients if they were not so depressed, or is also a cost win.
Yeah.
So I remember more a case I had when I did a study on severe dementia with another drug that affected, and then the staff came to me and said, "Bengt, we have really negative findings." What is negative? Yeah, I said, he's showing better cognition and being more active. Yes, but then he finds the way to Systembolaget. Could buy his alcohol. That was
Okay, that's a negative.
So that's the only negative I could find.
Okay, so showing initiative and really wanted to do something.
Yeah.
Thank you, Bengt.
Yeah, thank you. Thank you so much. Another open questions. How are your products protected patent-wise?
I can take that. They are protected well. If we look at NeuroRestore, we have patent protection, and it's all patented, and patent protection in the US is granted already as long as up to 2039. And then also why we are driving the new molecule Alzstatin is that, which we came with now in January, it's also because here we then can get a new patent and a long patent time. And that is also something which is very positive when it comes to competition, because many of the other projects that the competitors have are on fairly old molecules. So for us to have a longer patent time on our projects is very advantageous. So we feel very comfortable.
Thanks. Moving on to the last question here. Can 856 slow normal mental aging like memory loss and such?
It's certainly a good question. As Pontus mentioned earlier on, we see what we've seen in the animal studies is that we don't see effects in normal naive animals, but we have seen effect in aged animals. So where there's an kind of normal age-dependent cognitive decline, yes, we do see an improvement in memory as Pontus showed earlier on. So, but obviously the clinical trials here will be really guiding. But at least there's a potential, I think, for age-induced impairment.
If I can chip in there, this BDNF Val66 met polymorphism that we've discussed previously, that has actually been shown to have an effect in older adults by reducing the memory capabilities of these individuals without actually having been diagnosed with any disease. So there is a link between BDNF and age-induced memory impairment, one can say.
And we know, as Pontus alluded to, BDNF is going up or shown to be going up when you exercise. And we know that people who exercise get healthier. And if you get healthier, it's also very positive with regard to aging and longevity. So that is very positive. But of course, for us, this is just theory for the moment. So let's see what we can show in the future.
Thanks so much for answering our questions here. And I believe I will give the word back to you, Martin, for some concluding remarks.
Absolutely. Thank you, Ludwig. So concluding remarks, and I just need to do like that. So some take-home messages, concluding remarks. What we can see, which is so positive, is an increased interest and activity in the field of Alzheimer's. And this is both with regard to pharma companies re-entering the area, investing more, but also from investors coming in and wanting to fund projects. In Sweden, we have EQT, and we know that EQT have just launched a very big fund who are going to invest in dementia and Alzheimer's projects. Also, take-home message, antibodies are very promising, slowing disease progression, but they are not curing the disease and the patient's cognition. So what we can see is that there's a huge need for new, more effective, safer, and more convenient treatments. On the AlzeCure project, we can conclude that our projects are developing well. In NeuroRestore, this fall came with potential disease-modifying abilities that we have presented here, which is really add additional opportunities for the project. And on Alzstatin, our preventive therapy, where we are working towards that you shouldn't develop Alzheimer's disease, we have added a new additional compound, ACD680. And the advantage here is that it can have a long patent time. And we have also today shown the positive data on the molecule, which we now also have presented on several congresses. And if you want to learn more about our progress, I can recommend you to go to our webpage where we publish all our presentations, and you can learn more about our different projects. And then I can also say that please stay tuned. You know, we have a phase 2, which in our pain project, ACD440, which we are working hard on a readout. So follow us, and we look forward to also report on that in the coming months. So with that, I would like to thank you for attending today and for all the questions. And I also want to thank today's speaker, Professor Bengt Winblad, Johan, Sandin, and Pontus Forsell, and the whole AlzeCure team, and all our collaborators for doing great work. And stay tuned. We will come back